So as Carl said, tonight we're going to take a look at the immune system and what happens with COVID-19. Some of the latest information that's going on. I've been reading a lot of things about it lately, and still I was sitting in that chair a few minutes ago and looking on my notepad and found out something new that somebody figured out. So it's just constantly being updated. So, immunology. It's the study of the processes in your body that are going to help you to resist infection and diseases. Also, it helps with taking care of cancer. Extremely important. It's also extremely complex. And I'm not going to hit on everything tonight. It occupies a lot of books. New information is being discovered. When I originally studied immunology, was back in the early 80s, late 70s. A lot of it is completely changed from then. It's very hard to keep up with, actually. But I'm going to give you a brief overview of how your immune system works, how you're able to fight off diseases. The first defense of the immune system is called a passive physical barrier, as opposed to active. For example, your skin, your mucous membranes, those are always there and resistant to things getting to you. We have, opposed to that, the active immune system, and that's divided into two parts. There's the innate immune system and the adaptive immune system. The innate is non-specific. It doesn't recognize a particular antigen. It's not specific to a particular problem. It just recognizes anything that's not you. And it's an immediate response. It happens right away. Whereas the adaptive immune system is antigen specific. It recognizes a particular antigen and it reacts just to that antigen. It also has a lag time because it takes a while for it to be activated. But it also has a memory, so that later on, if you encounter the same antigen, then you're going to respond much faster to it.
Mr. Greninger, part of the slide is cut off in our view.
Okay, let me take a look at that. Continue, David.
Okay, so with passive physical barriers, the skin, the mucous membranes, it also involves a large number of chemicals. For example, there's defensins, there's enzymes. Some of them are on your skin. They're in your tears, your saliva, your breast milk. Immunoglobulins are also there, fighting off any invasive things. The pH of different tissues and fluids in your body also protect against invasion, because organisms have to have a certain pH to survive well. If an organism gets in your stomach, it's probably got a major problem, because you're producing hydrochloric acid in there at about a pH of 2, although it gets diluted by the food fluids that you drink. Your normal bacteria, all of us have bacteria within us, and flora. They protect us against invaders too. They compete with invaders. They don't give them room to be there. And that's true in your genital urinary system and in the gastrointestinal tract.
Rishi, are we solved?
Yep, we are.
Okay, continue, David.
Very good. Now that was the passive immune system. Now we're going to look at the active immune system I said that's divided into two parts. One part is called innate, the innate immune system. And the cells in that will recognize a generalized pattern of invaders. They look for things that are not self. Your cells that are yours have something on them called an MHC, major histocompatibility complex, that identifies the cell as you. Cells that invade you or viruses don't have that and get recognized as being foreign. And this leads to a generalized attack against any invaders. And also your own cells, when they become diseased or they're actually dying, then they will release things from inside of them, chemicals that will be recognized by your immune system. Your leukocytes are your white blood cells. There's a variety of white blood cells. It includes monocytes, macrophages, neutrophils, epithelial, dendritic cells. These are white blood cells in your blood that will recognize certain proteins on the surface of the pathogens. And that will cause certain proteins to be released by damaged cells. Now, if you recall when I talked about blood before, if you take a tube of blood and you centrifuge it, at the bottom you're going to have red blood cells and platelets. They're the most dense. That takes up almost half of the tube. At the top you're going to have plasma or serum. Which one you have depends on how you collected the blood as to whether or not the fibrin clotting factors got removed. And then in the middle, in a very narrow band, there's what's called the buffy coat or buffy layer, and that's the white cells. So even though it's a very small part of the blood system, it's an extremely important part of it. When a foreign cell is recognized by a white blood cell, by certain ones, such as the macrophage, then it will actually engulf it. It recognizes it from the chemicals on the exterior of it. It swallows it up, puts it inside a phagosome. Phag means to eat. And so it has a phagosome, which then combines with a lysosome. In your cells are little compartments, sacs, called lysosomes, that have enzymes in them. So the phagosome combines with the lysosome, and those enzymes that are in there will destroy anything that's in the phagosome. And that combination is called a phagolysosome. And it mentions there at the very bottom the MHC, major histocompatibility classes, that allow for recognition of whether an invader is foreign or not. This shows you the different white blood cells and how they're formed. So you can get, you'll see in the far left there, megakaryocytes that become thrombocytes. You have erythrocytes, which are red blood cells. And then you have mast cells. You have myoblasts, which then can turn into basophils, neutrophils, eosinophils, monocytes, and the monocytes can turn into a macrophage. When somebody does what's called a differential, they put a slide of your blood, a microscope slide, in the microscope and look at it, and they count how many of each of those they see. And that's a very easy way of determining some of the diseases or vectors that you might have. Your lymphoid progenitor can form a natural killer cell. It can form small lymphocytes, which then can become T lymphocytes. They're called T lymphocytes because they are made in the thymus. The thymus is where the T lymphocytes are produced. And they are very, very important to your immune system. It also forms B lymphocytes. Why is it called B lymphocytes? That's because in birds, it's found the B lymphocytes are matured in what's called the bursa of Fabricius. You don't have one of those. So yours just occurs where it occurs, pretty much in the bone marrow. And the B lymphocytes can in turn form plasma cells. And we'll take a little bit closer look at what some of these do. The main thing is what's in them and whether or not they can grab something and hold on to it, engulf it. By the way, the first time I gave a lecture to students was, oh, my mark. Okay. The first time I gave a lecture to students, I presented an hour-and-a-half lecture on what's called the complement system, part of the immune reaction. And I was very nervous, and I delivered that hour-and-a-half lecture in 15 minutes. And boy, were there a lot of questions when I got done with that. So if I go over something too rapidly or you have a question, please feel free to stop me and ask. The inflammation reaction, this is a very, very important part of the immune system. It's where it all starts. It's one of the first responses that you get when you have an infection. It's characterized by 4 qualities. First is redness. You develop a redness in the area. Second is swelling, as lymph and from the blood and the white blood cells that are going to be affected all swarm into that area, and it makes that area swell up. Another characteristic is heat. It'll be very warm. And the last one is pain. It hurts. So you get an increased blood flow and the release of a bunch of chemicals. There's ones called cytokines, there are eicosanoids, there's leukotrienes, chemokines, interferons, growth factors, inflammasomes, all these different kinds of chemicals that all have different actions. Very important.
David?
Yes.
On the slide with the diagram, what was the relationship between them that, what is the relationship between all of those and what makes the tree?
You have what are called stem cells that can differentiate. The earliest stem cells are called pluripotent. They can turn into any kind of a cell. This starts with the hematocyst, which is a blood cell. And it can differentiate into these different types to form all these different kinds of white blood cells and the red blood cell there too, as well as the thrombocytes. Does that answer? Okay, great. So here we have the diagram. You get a splinter in your finger. One of the things I hate the most. But when you do that, organisms are going to come in with it. And they can do very, very nasty things to you. So you get swelling in the area as those white blood cells come in. And as the lymph enters that area, it becomes warm, it becomes painful, and it becomes red. You get the development of redness. That's all the inflammation response. Your first defense. Oh, part of that got cut off. So this talks about the complement system. The complement system is involved in blood clotting and is a very important part of the immune system. It's also extremely complex. in the complement, it's a cascade. One thing happens, it causes two other things, causing three or four other things, causing 5 or 6 other things, and it just goes on and on and on. All these things happen. There's more than 20 different proteins that are released, and it forms a positive feedback system. As one protein is released, it causes the release of more, and causes more of its release of its own. The infects that you get from that include things as attracting immune cells to that area. It increases vascular permeability. So your white blood cells that are in the capillaries can get out of the capillaries into the tissues in order to attack whatever is needed. You get coating of the surface of the pathogen. That's called opsonization. For example, antibodies can coat the surface, and there are other things in there in that mix. You have an amazing variety of chemicals within your body. And that in turn ends up destroying the cell membranes of the cells that have invaded. This is the pathway for complement activation. And right down here, that is C5, which is called factor 5. Factor 5 comes apart to form factor 5A and factor 5B. Factor means we don't really have a name for it. And 20 years ago, even a little more, that's what I did research on. I did research on the turning of factor 5 into 5A and 5B. Very small part of this whole thing that has an amazing effect on the body, because factor 5A causes a lot of things to happen. So it's a very complex system. I'm not going to go into it in any more detail than that. So that was the innate immune system. Now we're going to look at the adaptive immune system. The innate, we said, was non-specific. This is going to be very specific. It's antigen specific. It recognizes a particular antigen and no other. The innate system had no memory. This system does have memory. It has cells that are going to remember that antigen and attack it much faster the next time it sees it. So you end up getting a much stronger response with this system. And it involves the T and B cells. The T cells are involved in what's called cell-mediated immunity. It's the cells themselves that are going to be doing the work. B cells are what's called humoral immune response, and it means the chemicals that those cells produce are what's going to do the work. And that's the immunoglobulins that we'll look at in a minute. Yes?
Some people are allergic to peanuts,
Yes.
and as they get exposed to peanuts over time, their immune reaction becomes more severe.
Yep.
Is that an example of this kind of immune system response?
Yeah, and actually that's immunoglobulin E. And we're going to look at the different immunoglobulins. That's
Carl-
what gives the
I can't
allergic
hear
response.
Carl.
Okay. Okay, there's a problem with my transmitter.
Oh, okay.
If I can-
Well, Carl's question was about people having a reaction to peanuts, and that is an allergic response.
Okay, I'm on now.
Okay.
Yeah, basically the question was, there are people who respond to peanuts negatively, and as they progress in this relationship with peanuts, over time, the response becomes more and more severe each
yeah.
time.
Hmm.
And so the question was obviously, is this a example of adaptive immune system response?
Absolutely. It is the memory cells, the B cells, that are recognizing that, and they're producing a special immunoglobulin called IgE, immunoglobulin E, and that's what's involved in the allergic response. To be honest, there's an awful lot about allergic responses that we don't understand. We don't know why it happens. We know there's a genetic component to it, but there's just an awful lot that we do not know. So I said that the T cells are involved in what's called cell-mediated immunity, meaning it's the cells themselves that do it. There's two main types of T cells. There are others, but those are the killer T cells and the helper T cells. And the killer T cells are just what you would think they might be, and they destroy things. As a matter of fact, they're very good at, if a virus infects a cell in your body, then that virus is taking control of that cell, and it's going to make it produce more viruses. Well, the T cell will recognize that, the killer T cell, and it will destroy that cell. Even though it's part of your body, it recognizes that the T cell of the virus needs to be destroyed before it can reproduce. Remember, viruses are not alive. They can't reproduce themselves. They have to use the cell's machinery. The virus, whether it be made of DNA or RNA, is going to go inside the cell and take over control of those processes. The T cells have to be presented with an antigen in order to activate them. That antigen it's part of the pathogen that's infected you, and it has to be processed and attached to a certain protein. Often it's the major histocompatibility complex, that one that identifies you, that tells this cell is self or it's not self. That's done in order to activate the T cell. Then the T cell, when activated, will find the associated infected cell, and it will release the cytotoxins that are within the T cell. It releases them, and it destroys the cell membrane of the invader. And that induces what's called apoptosis, which is cell death. It's especially important with, as I said before, the virus-infected cells, because it'll recognize that that cell's got a virus in it and needs to be destroyed before that virus can make copies of itself. The other type of T cell is a helper T cell. Now, it doesn't do that. It doesn't destroy the other cells. What it does is regulate both the innate and the adaptive immune responses. It requires a lot more interaction, a lot more time with interaction with that presented antigen in order for the helper cell to be activated. And the helper cells don't attack the cells, the pathogens. They instead release a lot of cytokines. And cytokines are very good at destroying things and cause a lot of problems, a lot of reactions. And the cytokines in turn will activate other cells, especially cells called macrophages. These are large white blood cells that move around as if they were amoebas, and they're very good at engulfing other things. Also activates killer T cells, which will destroy infected cells. This diagram shows the development of a T cell, the stages it goes through. It says it's intrathymic. Remember I said that the T cell is made in the thymus. That's why it's called a T cell. And it develops into the cell that's going to do the final activities. Now that's opposed to, that was the innate immune response. This is the humoral immune response. Your B cells will make antibodies. The antibodies are very specific. They bond to one particular antigen and no other. It's a chemical recognition. The antibodies are Y-shaped molecules, and they cover the surface of the B cell. When a T cell encounters the antibody-antigen MHC complex on the B cell, so it's the B cell that's going to present that to the T cell. And when that happens, it releases the lymphokines, which in turn activate the B cell so that it produces more antibodies. The activated B cell will divide, and it produces lots and lots of antibodies, floods the area with them. And they bind very specifically to those antigens, and that leads to destruction by activating the complement cascade. So a lot of different systems that work together to keep you safe most of the time. Some of those activated B cells will become long-lived memory cells. Stick around in your body. Some memory cells are there for life. Some only last a short time, but they will be able to produce antibodies to specific antigens. It'll rapidly produce extremely large numbers of antibodies when it encounters them again. The system is an adaptive immune response because it's an adaptation to infection in that individuals, excuse me, it's an adaptation to infection in that individual against that pathogen. Very specific. And this diagram shows the development and the activation of B cells. And again, no, I'm not going to go into that in detail. This is the sort of thing that you end up memorizing when you go to graduate school and study immunology. This is an antibody. Antibodies are produced by B cells. It's Y-shaped. Right here is a hinge area. This part is not flexible, and it attaches to the B cell. This part out here on the end is the antigen-specific part. It will be chemically designed to react with that particular antigen. And these, because of the hinge, they can flop around. And so if they encounter an antigen, they end up attaching to it, and you end up getting a lot of these antibodies attached to the antigens and activate other systems to come in and destroy them. There are 6 different kinds of antibodies. IgG is the main antibody. It's 70 to 75% of the immunoglobulins in your body, does most of the work of the antibodies in your body. IgD are similar in appearance to the IgG. They specifically activate B cells, basophils, and mast cells. The basophils and mast cells have a lot of different chemicals in them that they release and cause a lot of destruction. IgE, I mentioned a little bit ago, that's the one that's responsible for allergic reactions. IgA is found on your mucous membranes throughout your body. Your gastrointestinal system is actually not in your body. It runs through your body, but it's a tube that is really with the part of the exterior of your body. And so organisms can get in there, and that's why it's important to have the IgA on the mucous membranes. You also have IgA on your skin, you know, it's in nasal secretions and so on. IgM is the first one that binds with antigens, and it activates the complement pathway. Very, very important, because that complement pathway is very destructive to the pathogens. Now, I said there's 6 kinds, and that's because more recently, and it was quite a while ago, but it was after I took immunology courses, they found another kind of antibodies, and it's called cameloid antibodies. And they're called that because they were discovered in camels. So they decided to call them cameloid antibodies. And they are much smaller antibodies that circulate and will bind to, again, specific antigens. And so this is diagrams of them. As you can see, IgG, IgE, and IgD, they all look quite similar. The IgM, though, is a pentamer. It's 5 different Ys held together by disulfide bonds. And that means that when it ends up attaching to an antigen, you get a lot of attachment, a lot of activation. So it's very, very strong in the immune system. The IgA is the secretory immunoglobulin found in your mucous membranes, and IgA is a dimer. It's 2 of the Y-shaped immunoglobulins joined together. To keep your immune system working well, there's a variety of hormones, certain vitamins, sleep, rest, you need to get those. Repair, regeneration, those are all important to keeping your immune system healthy and working. I'm not going to get into any more detail on that in this presentation, though. It's going to take a while to look at some immune disorders. Times when the immune system goes bad, when it does things you don't want it to do. And like I said, sometimes we know why and sometimes we don't. Immunodeficiencies occur when the immune system cannot work properly. This can be due to a genetic inheritance. It could be mutations in the genes. Malnutrition can cause this. Obesity can cause this. Alcoholism, getting old can cause this. Or being very young. Newborns rely on the milk it gets from the mother, which contains the IgM antibodies, because the newborn is not yet producing any antibodies. AIDS is another one. And some cancers will make the immune system not work well. And surgical removal of the thymus. If you have your thymus removed, then you're not going to have T cells. As a matter of fact, there's a strain of mice that are called athymic. They're mice that are born with no thymus, so they have no T cells, and there's several things they can't do. T cells are involved in something called delayed type hypersensitivity, DTH, and so we use those mice to study the immune system when there's no T cells available. Autoimmunity is when the immune system can't tell you from non-you and ends up attacking the normal tissues in your body. Some autoimmune diseases include Hashimoto's thyroiditis, rheumatoid arthritis, diabetes mellitus type 1, systemic lupus erythematosus, and psoriasis. I want to talk a little bit more about some of those. Systemic lupus erythematosus. Lupus is where your immune system is attacking your own body cells. We, at one time, had a nurse working for us. She was one of the most wonderful people I have ever known. Young lady that my wife and I said we'd like to adopt her. She was taking care of our daughter, and she developed lupus. And hers was an unusual kind. Her immune system destroyed all of her fat cells. That's all it attacked, her fat cells. So she looked as if she were a bodybuilder, muscles standing out. But it also meant that she had no fat cells left in the pads of her feet. And so it was very painful for her to walk or even just to stand. And she ended up quitting, and we were very sad for that. Missed her. Psoriasis, I happen to have that problem, and the skin cells will turn a sort of silverish color, and they die and flake off. With some people, it's painful, it's bad itching. I've been lucky mine's not like that. There are creams that one can put on that to keep that from happening. There are medicines that one can take. They're advertised on TV constantly. The problem is it's your immune system attacking your cells, your skin. Why? Nobody knows. We do know it's stress-related, and that's why I shouldn't have been a teacher. But anyway, it happens, and it's actually destroying your own skin. You can take internal medicines that will make that better. However, those work by depressing the immune system. So then you're highly susceptible to getting other diseases. One of the biggies is they always ask, make sure your doctor tests you for tuberculosis before starting this drug. Okay. Hypersensitive damage. Hypersensitivities will damage your own tissues. There's 4 different types. Type 1 is called an anaphylactic reaction. This involves IgE, the allergic response, and it causes mast cells and basophils to degranulate, to release all those chemicals that are inside of them. And those chemicals cause a lot of things to happen. It can be a mild response, or it can kill you. It's associated with allergies. I happen to be allergic to penicillin. And when I signed up to get the coronavirus vaccine, I have to wait quite a while because they're insisting I get that at a place that can give emergency care if I happen to have an anaphylactic reaction. There's no reason to think that I would have one, but they want to play it safe. And that's a pretty good idea, really. The type 4 is one I mentioned earlier, has a 2 to 3 day delay in response. It's called DTH, or delayed type hypersensitivity. And one example of that is contact dermatitis. And this is something caused by T cells. So this is a man with contact dermatitis. Pretty bad case of it, actually. And it can lead to breakdown of the tissues. It can lead to a lot of swelling and such. And often it simply means you have to quit having contact with whatever is causing that. I had a cousin who went to work on a assembly line, and it turned out that the oil that coated the metal parts he was handling caused him to develop contact dermatitis. And so he had to quit that job. The cytokine storm. You've probably heard about this a lot recently because this is one of the things that's happening with COVID. In a cytokine storm, it's also called hypercytokemia, it can be end up having a fatal immune reaction. It's due to the uncontrolled release of a variety of chemicals, cytokines, chemokines, immune cell hyperactivation, causing massive inflammation, multiple system organ failure, and sometimes leading to death. And that's what is happening with the coronavirus sometimes. They're saying that that's what's causing the biggest problem. A cytokine storm can be triggered by various therapies, pathogens, cancers, autoimmune conditions, and monogenic disorders. We are not real sure why, we just know that these happen. Idiopathic inflammation. Idiopathic means we don't know what causes it. Well, there's not much I can say about that. It's an inflammation reaction, and we don't know why it happens. Now, the opposite of this would be immunosuppression. You can use drugs to shut down part of the immune system. Especially useful when you have transplant tissues. Transplanted tissues coming from another person are going to have a different MHC, major histocompatibility complex. And so it's going to be recognized as non-self. And your normal immune system is going to attack it and destroy it. That's one of the reasons that they say it's best if they can get like a brother or a sister as a donor, because they get somebody who's very similar so that there's less recognition of the antigen. I had a close friend who was one of the world's first successful liver transplants. And after that, she was put on some really strong medicines to control tissue rejection. One of the potential problems with that is developing something called Stevens-Johnson syndrome. Now, normally in Stevens-Johnson syndrome, you lose 10 to 30% or more of your skin. It goes away. People who have that end up being put in a hospital, in a burn ward, and treated as if they had been badly burned. And often it results in death. In the case of my friend, it was 100%. All of her skin went away one day. Because that is so extremely painful, she was put into a medical coma. There is no cure. There is nothing that can really be done for that person. And because my wife and I were friends of her, and because my wife was a physician, the nurses and doctors asked my wife to talk to her family, because the family were keeping her on life support there. And my wife got them to see that she was not the kind of person who would want to stay on life support and have no chance. And so they finally took her off and allowed her to pass. The opposite of this, there are people who claim they can make your immune system stronger and better. And this is a claim often by chiropractors, homeopaths, acupuncturists, some of them, that they claim they can improve the immune system. And the truth is, there's never been any evidence to back up any of their claims. Homeopathic doctors. I have a whole lecture I could give on that. Vaccination. In vaccination, an antigen from a pathogen is used to stimulate both the T and B cells in order to produce the long-term memory cells that will be able to fight an infection in the future. The antigens cannot cause the disease, but because they're only part of the pathogen, the immune response is not very strong to them. So they have to add another chemical. It's called an adjuvant. And that adjuvant increases the effectiveness of the vaccine. Smallpox has been completely eliminated. There's none in the world population. There is supposedly 2 labs in the world that have some smallpox kept in them for purposes of research, and that's it. Polio and measles were coming very close to being eliminated also. However, because of the anti-vaxxers, it's now become a problem again, both of those diseases. And this is my own personal opinion here, but I'm really against anti-vaxxers. It's my experience they're usually misinformed, or they consider an anecdotal piece of evidence more important than statistics. My sister is a leading anti-vaxxer, and she knows of a case where somebody got hurt by the vaccine. Well, that person might have been hurt by the vaccine, they might have been hurt by something else, but they had a much better chance of surviving by having the vaccine. It is sometimes possible to get anti-vaxxers to understand the truth, but that's not been my experience, especially with my own sister. I'm not allowed to comment on her postings on Facebook anymore. Okay, so I've talked about the immune system. The thing I want to talk about next is COVID-19 itself. But I've been talking a lot, you've been sitting there a lot, and people at home. So I want to give you a 5-minute break, stand up, move around, relax for 5 minutes, and we'll start up again and talk about COVID-19. That went a lot longer than it did at home. I might end up taking the whole time. Sure.
Okay, well, it is what it is.
Yeah.
Is this helpful? You guys are going into medicine, you know. So.
Well, and even for people that aren't, it's been so long since I've taken biology. I took it freshman year, but I know I'll have to take it again in college. So this is really, really helpful to remind me of things. Also, I'm really excited to learn about COVID more.
Well, it's definitely it's an appropriate time for it. But a lot of the stuff that I've gone over here, you wouldn't get in a high school course, even in Bio 2. Don't go into this much detail. You would get it if you took immunology as an undergraduate student. And then if you went into a medical field, you'll get it a lot.
When you consider the complexity of a human body, it is an absolute marvel, especially the immune system.
Yep.
It is absolutely, you talk about the probabilities that certain things could have occurred by simple random mutation and natural selection. And some of those probabilities reach orders of magnitude in 1 times 10 to the minus 74 chances. And while in statistics we cannot use the word impossible, we do get to the word preposterous. I think you know where that goes. David?
Yes. Yeah, my wife always said that it's when you look at all the things that can go wrong in developing a baby, and yet so many of them are born perfectly. That's definitely a true miracle. And she was both a family practice doctor and a medical geneticist. So she knew a lot about it. Well, it hasn't been 5 minutes, but nobody actually seems to be taking advantage of it.
No, they're racing. We're talking racehorses here, ready to go.
Well, I'll tell you, in college, in high school, they kept emphasizing to us, never talk for more than 15 minutes. That's your attention span as a high school student. And that's for the smart ones. And then you get to college and you find out each lecture is an hour and a half long. Yeah. So, okay. So, if nobody objects, people at home, I'll go ahead and start up. So we're going to look at COVID-19. And we have these wonderful diagrams that people have made showing the idea of what the COVID-19 virus looks like. The virus is actually called Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. It's the disease that is called COVID-19. And unlike some of the congressmen and a former president thought, no, it's not because there were 18 diseases before it. It's 19 because it first appeared in 2019. Okay. The disease first appeared in Wuhan, the Hubei province in China. That occurred in December of 2019. Its first known appearance in the United States was in March of 2020 in King County, in the state of Washington. It is highly contagious, and it has caused a worldwide pandemic. The virus has DNA. Actually, I shouldn't have worded it that way, because it's not DNA, it's RNA. I'm sorry. It has messenger RNA. It's 90% the same as the messenger RNA of a coronavirus found in bats. That's why it was originally believed to have derived from the open air market that exists in Wuhan. There's a huge seafood market there, and they have bats hanging up, and the blood drips onto other things. And so, because it's the RNA sequence is extremely similar to the bats, so I thought it came from the open air markets. Also, the earliest cases of people were people who were either shopping or working in that market. So it was a reasonable deduction. However, most vaccine specialists now say it's probably a 90% chance that it was actually developed at the Virus Research Center in Wuhan. And it either accidentally or purposely was released into the city. One of the reasons is because it has so many different activities, so extreme, that's indicative of an engineered virus. Wouldn't expect that in a normal virus. And that's one of the problems with biological weapons. If this happens to be one, it's that they're usually non-specific and they can infect anyone. And so your weapon often turns back on you. Similar to what Haber found with the chemical warfare that we talked about a long time ago. A great man who developed fertilizer that saved the lives of millions by allowing us to produce much greater amounts of food, and then also developed the first chemical weapon to be used in warfare. And the first time he used it, the wind blew it back on his own troops and killed them. Coronaviruses are very different from the flu virus. The big trademark for the coronaviruses are those spikes that you see on them. There are 7 known coronaviruses that infect humans. One of them is the MERS-CoV virus. It's a beta coronavirus that causes Middle Eastern Respiratory Syndrome, also known as MERS. And then we're dealing these days with SARS-CoV-2. There was a SARS-CoV-1, except they didn't put the 1 on it because it was the only one at that time. So SARS-CoV is a beta coronavirus that causes severe acute respiratory syndrome, also known as SARS. Symptoms of COVID-19: fever or chills, either way, or both. A cough, shortness of breath, difficulty breathing, fatigue, extreme tiredness, muscle or body aches, headaches, a new loss of taste or smell. I like that they put that new in there because I lost my sense of smell about 30 years ago. A sore throat, congestion or a runny nose, nausea or vomiting, and diarrhea. You could have one or many of these symptoms. And of course, we know that you can be asymptomatic and have no symptoms at all and still have the coronavirus. Some predispositions. These are factors that make you more likely to get infected, to develop a more acute symptoms, be a lot harder on you, or even to die. That includes being elderly, being immunocompromised, your immune system's not working, having heart disease, having had a heart attack, having problems with the heart, obesity, type 2 diabetes, chronic kidney disease, COPD, which stands for chronic obstructive pulmonary disease, means you have trouble with breathing. Recently, they found that Down syndrome children are much more susceptible to having a bad infection. Pregnancy is a predisposition. Sickle cell disease and smoking. Don't smoke. Don't ever smoke. Not worth it. I like this chart because it talks about the cold versus flu versus coronavirus. How do you know which one you have when you get sick? And it can be a little hard to tell, but one of the main things is it's going to be it can be much more serious. Put you in the hospital, which fills up the hospitals, and then there's no place to put people to treat them. You can find a lot of these charts on the internet. All I did was type in cold versus flu versus coronavirus, and that popped up, along with several others. SARS-CoV-2. The virus is a single-stranded messenger RNA, 29,891 bases, that encodes for 29 different proteins that are used by the virus for infection, for replication, and assembly of the virions, the particles that make up a virus and then get put together. The spikes on the outside of the virus bind to human angiotensin converting enzyme number 2. And because they bind with that and it's on our cells, then that allows it to fuse with the cell membrane and then enter the cell. And there are more than 90 recognized genomes for the SARS-CoV-2 virus. One of the concerns with the mutations that are occurring, many of the vaccines being developed, and we'll take a look at those, work against the spikes on the outside of the virus. And those may be changing with mutations. And that means that some of the vaccines wouldn't work and would have to be modified. We don't know. The virus is commonly transmitted in aerosolized droplets. However, recent evidence suggests it can be transmitted as viral particles in the air. We know it can be transmitted at surfaces, but it's actually been found to be much more likely that it would be transmitted in the air. Most infected people are actually asymptomatic, have no symptoms. But they can transmit that virus to other people. It's not yet known if people who have recovered can still transmit that virus. So, recommended methods for avoiding the virus. By the way, I love that mask. I want one of those. If you can't read it, it says it goes over your nose. And I'm tired of seeing people with the mask on their chin, or just over their mouth, or even on their neck. And they say, well, I'm wearing a mask, that's all that's required. Properly fitted this face mask, or even two of them. It's now being suggested that you wear two. Washing hands thoroughly with soap. Sing the happy birthday song twice. Keep hands away from your eyes and your mouth. Social distancing, 6 to 10 feet. Now everything I had read says 6 feet, but I had a repairman come to my house the other day, and they sent me a message ahead of time that I am to stay 10 feet away from him. Okay, sounds good to me. Clean and disinfect frequently touched surfaces. Avoid crowds, and especially avoid poorly ventilated areas, because that's how it gets around. And of course, the suit I'm wearing is a very good way of preventing getting coronavirus. And there are many, many posters of this that you see all over the internet and now being posted in stores, mentioning these things that we just talked about of how to keep from getting infected. So some brand new statistics. Worldwide, 105 million people have come down with COVID-19. 58.5 million of those have recovered from it. And 2.29 million have died worldwide. It'll be 3 million soon, I'm sure. In the United States, 27 million have come down with COVID-19. Below that, it says plus 127,000. That's because yesterday it was 27 million, and today there was another 127,000 cases. It's still happening. Apparently, nobody kept records on who's recovered because there was nothing there. But deaths, 456,000 as of yesterday in the United States. And another 5,116 died today. In Washington State, 322,000 cases with an additional 2,438 today. And deaths, 4,492, and 26 more died today. In King County alone, 79,342 people have come down with COVID-19, and 279 more today. And 1,270 people have died in King County from COVID. And today, 4 more died. It's getting to where almost everybody knows somebody who died from COVID. So why is COVID-19 so deadly? Well, one reason is that cytokine storm that we talked about before. This virus causes the immune system to release many different chemicals in your body that end up causing multiple organ system failures, leading to death. One thing we do not yet understand is long haulers. Some people will recover from the virus, but their body systems don't recover, even among previously healthy, active young adults. Saw a program yesterday on TV about a young lady who was a runner, was hoping to make it to the Olympics eventually. And she can't walk now. She's in a wheelchair. She's recovered essentially from the COVID-19, but it has destroyed her muscles. And this is happening to more and more people. And this was a young lady who was very healthy, extremely active athlete. And we don't know why. That's part of that engineered virus that I was talking about.
So, David,
Yes.
first of all, can you hear me?
Yes. Softly.
Yeah, your mic's not.
No, I don't. We can hear you because we're in the same room, but I don't think it's through the mic.
Okay. Hello, check. Yeah, okay. So the girl's muscles were destroyed. Obviously, it wasn't COVID that destroyed that. It was her immune system destroyed her muscles.
Yes.
Okay.
That's
So-
the cytokine storm.
Okay. And so what we have here is a secondary reaction going on that's doing the major tissue damage
That's right.
that we see with COVID.
And it's believed that's what all the deaths really are. That it's the secondary things that happen because of having COVID, that it's destroying other things. Currently, there are more than 90 different attempts being made to develop a vaccine for COVID-19. Those vaccines fall into 4 broad categories. And all of this is taken very liberally from Nature. Nature magazine had a great article on this, and so they do much better charts than I can. So, the 4 types are virus, either inactivated or weakened, viral vectors, either replicating or non-replicating, nucleic acids, either DNA or RNA, protein-based with protein subunits, or virus-like particles. And it shows the number of different vaccines, how many for each one that are in development. So for the virus vaccines, there are currently 7 teams working on vaccines using the whole virus. Either it's been weakened so that it won't cause the disease, or it's inactivated so it can't cause the disease. You can't say it's killed because it was never alive. It's a virus. This is how vaccines were developed against measles and polio, and they work quite well. And in this diagram, it shows the different stages for doing that. On the left, you have the weakened virus so that it can't cause the illness, but it will be seen as foreign and cause the development of B cells and immunoglobulins that are specifically designed for that. And then the inactivated virus is going to be even less of a chance of doing that. They either destroy it with a chemical like formaldehyde or they heat it up. Salk and Sabin polio vaccines. This one's kind of dear to my heart. When I was a little toddler, Salk and Sabin, at the same time, both came out with polio vaccines. The Sabin vaccine was a weakened virus, and it was a liquid that you drank, I think. And the Salk vaccine was an inactivated virus. Salk felt it was very important to not use a weakened virus because there would be a chance that it wouldn't be weakened enough and would still cause the disease. Unfortunately, the manufacturer of the Salk polio vaccine let a batch get through without inactivating it. And all the kids that were treated were given active virus particles, and it caused those kids to develop polio. Very, very sad thing that happened. It was not Salk's fault. It was a manufacturing error. I can remember standing in lines to get each of those vaccines. My dad wanted to make sure that I got them, and I grew up knowing some people who had polio and were crippled by it. The viral vector vaccines. 25 teams are using that approach. A virus is changed genetically so that it makes the virus's proteins, but it is weakened so it cannot create the disease. One type can reproduce itself, the other cannot. So those are the two different types of viral vector vaccines. The idea is that even the one that can reproduce itself, it's weakened enough that it can't cause the disease. And then this diagram shows both parts of that, the weakened one on the left and the non-replicating, well, they're both weakened, but the one on the left is replicating and the one on the right is not replicating. And injected into a person to develop the immune reaction. The third type are the nucleic acid vaccines. There are 20 teams that are investigating that. They either use DNA or RNA. That codes for the virus, and then it's inserted into a cell so that the cell then makes a particular protein of that virus. Usually it's the spike proteins. And both the Pfizer and Moderna vaccines that are now approved for use in the United States are that kind of a vaccine. They're both using messenger RNA. There's two more that may, or probably will be okayed fairly soon. Johnson & Johnson and AstraZeneca. And those have different activities, different types. And the diagram for nucleic acid vaccines, whether it's using DNA or the RNA, it gets injected and you end up creating the proteins that are the antigens that the antibodies will recognize.
David, the Johnson & Johnson, I understand, is only about 60% effective.
Yes, but it is more effective at not at keeping from having serious disease.
So those who have been inoculated stand a higher chance of contracting it, but a smaller chance of succumbing to it.
Yeah, exactly. And I'm going to talk a little tiny about that too.
Okay.
Protein-based vaccines is the fourth kind. There are 28 teams that are using that approach to developing a vaccine. In that case, they use fragments of proteins that the virus makes, or even parts of its protein shell that coat the outside of the virus. And those are injected directly into a cell to cause an immune reaction. And there's the chart that shows it. On top is the protein subunits, and on the bottom are virus-like particles. And this is to answer what you were saying, Carl. And this is latest research I found. This came out in February. Some researchers, in particular, some in Oxford, at Oxford and London, they're looking into mixing vaccines, which could increase their effectiveness, especially if the vaccines have different modes of attack. And so combining them might give much greater results. That was essentially done with the polio vaccine in the 1950s, when everybody was taking both the Sabin and the Salk vaccines to try to have a better chance. That same approach was used against Ebola and HIV, mixing different vaccines to get greater effectiveness. There are those who are worried about safety of the Pfizer and Moderna vaccines, and these are some justified worries. It's believed that the two currently available vaccines are safe, but we don't have the results yet from long-term studies. They are ongoing, so we are taking some risk if we take the vaccine, because it could end up turning out later that there is a problem. We just don't know for sure. There is no indication yet that there's any problem. They also need to study to run studies of the use in young people, because they don't know how it affects adolescents and children. And studies need to be done with pregnant women to make sure that it's not causing harm to the fetus. And the big problem right now that everyone's talking about is mutations. Viruses reproduce in vast numbers, very, very rapidly. So we can expect to see mutations. They're going to happen. Normally, the mutations that succeed will make the virus better at infecting cells. They do not make the virus more lethal, because a dead host is of no use to the virus. They're not going to be producing more viruses. However, previously I have read that the mutants are more lethal, and today I read that the mutants are not more lethal. So I honestly do not know at this point. And it just may be one of those things that we've got to figure out yet, because we do have them and they are spreading, and they definitely are more contagious. Everybody's agreeing on that.
I understand the South African that's made it into the United Kingdom is initially thought to be certainly more communicable, and there was an article earlier this week that it may be more lethal as well.
And I read that it was either yesterday or today, Australia went into complete lockdown because they had one person who got sick with COVID-19.
Ah.
They have been so effective, which some people say, well, of course they're effective. They're an island. And they're only populated in certain coastal areas. Yeah, but still, they seem to have done. The other thing is, New Zealand also has done quite well, and both of those countries are run by women. That might suggest something. My wife insisted that they were going to end up developing a way to clone women so they wouldn't need men anymore. They'd keep a few of us around for playthings. I figured I wasn't going to be around. Okay, the mutants of the virus causing COVID-19 seem to be developing both. That's what I heard up until today. We do not yet know if the vaccines that we have, the ones we already have approved, if they're going to need to be adjusted to handle the new variants. But the manufacturers are saying they think it would be a simple matter to make adjustments. It's not like starting from scratch. There's a lot we don't know about these vaccines because that's proprietary information. So researchers aren't allowed to find out how those vaccines are made, the specifics. Major problem right now is vaccine distribution. Because until quite recently, there was no national program for distribution of the vaccines. None, nada, nothing. It's hoped that problem will be fixed very soon, because our new president is putting an emphasis on this. He has put COVID-19 as the #1 problem that he wants to work on. Read recently, in 1912, New York City was hit by a flu epidemic. They managed to vaccinate 9,000 people in 1 month, in one city. In 1912. All right, the last thing I'm going to talk about is a BLAST. BLAST stands for Basic Local Alignment Search Tool. A BLAST finds regions of similarity between biological sequences. It can be in proteins or in DNA, either one. The protein compares nucleotide or protein sequences and calculates the statistical significance. For example, you're doing some research on mice and you find that there's a part of their genome that codes for a protein that makes them able to resist a certain kind of cancer. And so you wonder, well, I wonder if there's a similar protein or similar genome in humans. And that's what a BLAST does. It allows you to check for that, to find similar sequences. It is one of the newest tools and a very important one. One scientist said it's like the Google search of DNA. So the website is blast.ncbi.nim.nih.gov. So that's National Institutes of Health, the main government organization that funds biomedical research in the United States. The program is run by the U.S. National Library of Medicine. It is a tool that is used in a subject area called bioinformatics, and that's become extremely important. So I'm going to give you a long-term assignment, very long-term. I want you to begin doing this assignment, but it's going to take a very long time. It's going to be worth it, because you're going to learn how to use a BLAST, and that's an important thing. You will go through a tutorial on how to do a BLAST. There are questions at the end of the assignment. Eventually, I will want you to answer those questions. Now, this is an assignment that is usually given to college grad students. But I'm sure that you guys are going to be able to do it. okay? It's just, for them, it's a 1-week assignment. And I don't expect you to have this done in a week, okay? It's lengthy. The assignment is at this website, community.gep.wustl.edu.
And we'll send that out by mail.
Right. And the title of it is Lab Week 8, an in-depth introduction to NCBI blast. So eventually you will answer the questions that are at the very end of the assignment. But it's going to take a while to get through the thing. And give credit, the template I used came from SlidesGo.